Now Deep Thoughts by Jack Handy.
No, it's Transport Proteins by Dr. Malone. I don't know what that's about. This little tutorial is going to be about transport proteins. There are three main types we're going to talk about. Channel proteins, carrier proteins, also known as transporters, and pumps. Okay. Both the first two, channels and carrier proteins, are passive transport. So what does passive mean? Diffusion. What is diffusion? Always traveling from high to low concentrations. No energy required, right? So that is the first two. The third one, the active transport, anytime we say active, it means ATP, needs energy. So if something requires energy, that's usually because it can't go by diffusion, right? If you can go down your concentration gradient, there's no reason you need to add energy. So active transport requiring energy must be low concentration to high against concentration gradient. So those are the three main types. We're going to discuss channel and carrier proteins first, and then we'll deal with active transport. Again, all of these are types of transport across a membrane. Ion channels, or channel proteins, are exactly what it's described. To pass ions. What are ions? The charged stuff. What did we learn when we talked just about diffusion and membranes? These guys don't cross membranes well at all. They're at the bottom of that chart. Does not cross. Charged stuff can't get through what? What can't they get through? The hydrophobic regions. So in order for these to pass, we need specialized membrane proteins to make a channel, right? Make a passage through the stupid membrane, right? If there's your membrane, right, you need these proteins here on the edges to essentially cut a hole in the membrane so these bad boys up here can go right on through. And they will move on their electrochemical gradient. It's just a fancy word for a gradient for charged stuff. No big deal. So if there's high concentration on one side of a plasma membrane, and let's say it's this side, and these are sodiums, they're going to go which way? Boink, that way. If there's a higher concentration on the other side, they're going to go the other way. They will diffuse through their channel. That means down their concentration gradient from high to low. And if it's diffusion, what does that also mean? You got it. No energy required. Oops, can't even spell energy. So here's a picture, and this is just showing you the concentration gradient. It's not showing you anything else. So if these guys were to pass through this membrane, can they pass through the way it is? No. They'd have to have some kind of channel right here. These proteins would have to be on this side, channel proteins, in order to let these guys go through. And then we'd see sodium, high. What about chloride? Sodium's going to be net going this way. What about chloride? Chloride's going to go this way. Chloride's going to go that way. Chloride is already at its equilibrium. So there's going to be no net flow. The concentration gradient here we see is the sodium. So here, sodium, way more up here than down here. They're going to flow until they reach equilibrium. And then they'll go back and forth, right? They don't stop moving, but there's no net movement. Took a snapshot at any given time, would look exactly the same. And that's the deal with this guy's going this way and that guy's going that way, but there's no net change. At any given time, it's at equilibrium. Channel proteins are selective. Ooh, what does that mean? It just means that each one has a structure that allows only one type. So there's a sodium channel, a potassium channel, a chloride channel. There's not one channel for all the ions. Every ion has its own channel. Only lets that one through. So you can, even if there's an electrochemical gradient of one versus the other, sodium's high on one side, potassium's high on the other, there's a sodium channel, there's a potassium channel, okay? And they can determine which one gets to go. This one would want to come this way, and this one would want to come that way. Would have to. It would, right? There's a higher concentration here than here. There's one here, none here. Boink, he's going to want to do that. One here, none here. Boink, he's going to want to do that. If the channels were always open, it would just flow on its concentration gradient. As it turns out, the channel proteins don't always have to be open, and that can help control when the ions flow in and out. It's not just going to happen willy-nilly on the concentration gradient. If they all went through the same one, there'd be no way to control. It would just be by diffusion on concentration gradients. There'd be zero control over it. That would be a huge problem for lots of cell types. The main one is neurons, where there's potassium and sodium channels, and flowing across the membranes is what generates that electrical impulse, that active transport down the axon, okay? So what do they do to control this willy-nilly diffusion that happens down concentration gradients? Well, many of the channels are actually gated. They can open or close. So they're not just always this passage that's open. Sometimes they're closed and they won't let things in or out the other way. And that makes sense. So how does something open and close? That sounds like it's doing work. But by definition, what did we say about these channels? They were diffusion. No energy. So obviously the open and close does not use ATP. So what happens? Well, these gated channels work by when there's a binding of their molecules, so sodium or potassium, usually whatever the channel is for, it can change the conformation of the protein. Remember, protein function is all about shape and conformation. Conformation or shape in a protein is all about the chemistry, right? If you bind something that's positively charged or negatively charged to a protein, that's going to change the interactions between the R groups of all the amino acids in that protein. It can change the conformation without using any energy. And so that's what happens. Gated channels just change their conformation based on interactions with their ions. And that allows for this flow of them by diffusion, but controlled. Okay? So we call that facilitated diffusion. It's helped by something, a gated channel, right? In this case. So again, it's still passive, no energy. It's powered by this electrochemical gradient from high to low diffusion. It's just sometimes we can block it from going through because it's not just a hole, right? If it was just a hole that was unclosable, it would just flow on the concentration gradient as it would. But because you can think of it as a gate, then if you have your channel protein and think of it as a little lid on it, right? And the lid can open or it can close. So if it's closed and there's a whole ton of these guys on this side and none on the other, they're not going anywhere. Once it pops open, then bingo, they can all flow through. So that's how gated channels work. Again, passive, no energy, facilitated diffusion. Here's our little picture. So here we have it closed. This guy down here is closed, right? This is outside the cell. This is inside the cell. As soon as it opens, then this is the potassium channel. All the potassium can flow this way. So they're saying, right? All the potassium's on this side, right? When it's here, that causes the gate to open, right? And potassium will flow down its concentration gradient, right? We'll say there's 1 up here. From high to low. Concentration's high of potassium to low. Guess what happens when it reaches equilibrium? It probably closes. We don't have that pressure to open by all of these guys on this side. So that's how the gated works. So it just doesn't always flow little by little, right? It has to build up out here, then open and cross through. Like I said, there's a different one for every single type, and these channels do facilitated diffusion. Anytime diffusion is helped or facilitated, right? What does the word facilitated mean? What's our PLF? Peer Learning Facilitator. Something that helps out. Doesn't do the job for you, but it helps diffusion. Diffusion is going to happen because it's high to low. But it has to be helped by some sort of protein because ions can't just cross by diffusion. So we can't just call ions crossing membranes diffusion because they need help. They don't just cross the plasma membrane. What kind of help do they get? They get channel protein or gated channel protein help to facilitate their diffusion. No energy because it's diffusion. Facilitated diffusion can also go by carrier proteins or transporters. So the channels and the carrier proteins are both facilitated diffusion. Yeah, it is a little bit scary, but that's all you have to remember. Facilitated diffusion is diffusion just helped. So a carrier protein, again, no energy. Channels, no energy, but they help out diffusion. So what is a carrier protein's job? How is that different than a channel? Same idea, down a concentration gradient, but in this case, it talks about anything other than ions. So other than ions that need to pass. Okay, so our example would be glucose. Remember, we talked about glucose not being able to pass through just the phospholipid membrane. We used those examples. That was our big guy, our solute that couldn't pass, that caused osmosis to happen. So glucose doesn't go through a channel protein because it's not an ion. It's a big molecule, right? It's a sugar molecule. So what does it have to use? It has to use a carrier protein to go through. So all big stuff that crosses our membrane has its own carrier protein. Just like the channel proteins that have one channel per ion, a different carrier protein or transporter per molecule. So any kind of large molecule that needs to pass has to have its own carrier protein. Because the lipid bilayer barely permeable to glucose. Remember that chart? And so it needs a glucose transporter. It's named GLUT1, and it increases membrane permeability to glucose. It allows glucose to pass through. It facilitates the diffusion of glucose from high to low. Here's our little picture. It's the hypothesis of how it works. Look, what's up here? Ooh, a high concentration of glucose. What's down here? Ooh, a low concentration of glucose. Can glucose just pass through the phospholipid bilayer? No, it totally sucks at getting through there. But if there's a carrier protein here, a transporter, the concentration of glucose here can bind. Ooh, glucose has chemical groups in it, and so do proteins, and the interaction of the chemical groups in glucose and the R groups in the amino acids change the shape of this protein. That sort of causes this part to squeeze together and this part to open up, and boom, we poop out some glucose. Okay? High to low, no ATP. High to low, no ATP, uses a protein. What do we call it? Facilitated diffusion. Yay! Okay, active transport.
Hee-haw!
Yep, it's the different one. This one is active, means yes, ATP. And if you need energy, that means it has to be going from low to high against its gradient. Otherwise, it wouldn't need ATP. It would just flow down its concentration gradient. So it's extremely important to know this. Active transport. We have to do something, provide energy. We call it a pump because we're pumping it out, right? Or pumping something in. Pumps require energy. They don't just happen. It's not just pouring something out. You have to pump it against something. So a sodium-potassium pump uses ATP to transport an ion. What? I thought that was a channel protein. Dr. Malone, you're out of your mind. Ooh, but what is the key? Against their concentration gradient. What would we call it if it was with concentration gradient? Yes, we would call it facilitated diffusion. Right? And what kind of proteins help out? Channel proteins. Look, it's active transport. High concentration of sodium, right? And potassium. So in the case of active transport, there's a high concentration of sodium on the outside of the cell, a lower concentration of sodium on the inside, and so if it were just facilitated diffusion, which way would it want to go? This way, right? Down its concentration gradient. If the cell needs to pump sodium ions out, it has to provide some energy. It has to have a transport protein, an active pump protein, and needs ATP, right? To get these small amounts of sodium to pass out of the cell where it's even more, right? We're going here from low to high. That's against a concentration gradient. And it needs, gradient. It needs ATP, active transport. Active transport. So as a summary of membrane transport, we have three main mechanisms of membrane transport. We have diffusion, facilitated diffusion, and active transport. What are the two types of facilitated diffusion? Channels, right? And what else? And transport proteins. Ta-da! What was active transport? Right, that was our pumps. Passive is always diffusion or facilitated diffusion, right? No ATP. No ATP. Right? Active transport, yes, ATP. This is high to low. This is low to high. A picture to help us understand this. Right? Here we have active transport, facilitated diffusion, and diffusion. Diffusion, right? Nonpolar small things just going in and out on their high to low concentration gradient. Again, very passive. No proteins involved. Right? What about this? Facilitated diffusion. We can have you guys fill this out. And this would be a nice little homework problem to screenshot and upload. Right? What is this? What type of transport is this? What we didn't really talk about, the water, so let's ignore this one. Okay. And then what is this when it's an ion? So pretend this is glucose, pretend this is an ion, and then tell me about active transport. What is it the active movement of? Right? How does it go on its concentration gradient? And what proteins are the names that they're involved in. So fill this out, take a screenshot and upload. So please, to 11.8, transport homework, take a screenshot and upload. This is under February 18th. A little more review, and some key concepts. So thanks so much, and I will see you soon.